Hello student, welcome to EPG Pathshala. I am Dr. Ajit Jaswal from Department of Anthropology, Pondicherry University. Today we are going to discuss about a very important module, that is Introduction to Genetics under paper Physical and Biological Anthropology. Dear student, in this module we are going to develop and understand about what is genetics. We also have a brief idea about the structure and function of cell, and also have a comprehensive knowledge of DNA structure and its function. We also try to know the basic concept of chromosome and cell division. Let me give you a clear-cut information about genetics. The question that often came in our mind is why we are morphologically similar with our parents and different from member of other families. In the same way, there is another question, why member of the population group share some resemblance with each other and differ from those of other population group, or why such similarity and variation do exist among individuals. Is there is something that we inherit from our parents from the time of our birth, or do we share something with the member of our population group? The result in having similarities morphologically. Yes, we do inherit something from our parents that make us look alike with them. That sometime or something came to be known as gene. The word gene was first coined by Johnson in early 19th century. Without knowing the biochemical properties, but just because it is nice small word to describe something. And it was later on Bateson who defined heredity as similarity due to relatedness. The basic nature of the gene was described by an Australian monk called as the father of genetics, that is George Mendel. Gregor Mendel, more than a century ago, after a careful experiment conducted in a common sweet pea plant or called garden pea between 1856 and 1868, he summarized his finding in two laws. The law says that the gene was recognized as a particular factor. It is not something that passes unchanged from parents to the progeny. He described that each particular factor or gene exists in pairs in individual organisms. When an individual has two different particular factor or gene, only one of them expresses, and it is so said to be the dominant to the other, who is said to be recessive. During the gamete formation, the paired particulate factor separate or segregate randomly with equal likelihood. This is known as Mendel's law of segregation. The second law is known as Mendel's law of independent assortment. In this law, he described that during gametic formation, segregating pair of a particulate factor is short independently of each other. That is, each paired gene freely separated and do not influence the separation of other paired gene during gamete formation. These two laws explain the basic fundamental principle of inheritance. However, his groundbreaking research was discovered only in 1900 by two independent scientists. One is William Bateson and Hugo de Varis. 16 years after the death of Mendel and 35 years after he published it. After the discovery of Mendel's idea of inheritance in 1900, a whole new science called genetics, and Bateson coined the term in 1908, emerged. So the genetics is that branch of science which deals with the study of inheritance of unit factor or called gene. These genes are present inside the nucleus in the form of DNA in every cell. Cell. Cell is the basic unit of every life. It is a microscopic organism, organic body which carries genetic material and other organelles required for cellular function. All living organisms are made up of cells. Some are made up of single cell and called as unicellular organisms, like bacteria and protozoans. This single cell performed all the activities of being alive. Other, including human, are made up of multiple cells and hence called as multicellular organisms. Among these organisms, different activities are performed by different specialized cells. All the living organisms on earth can be divided into prokaryotes and eukaryotes. Prokaryotes are those organisms with no major compartment within the cell to separate the genetic material from the other component of the cell. Bacteria, blue-green algae are some of the examples of prokaryotes. Prokaryotes are the earliest form of life on the earth, appear in the fossil record of about 3.4 billion years ago. On the other hand, eukaryotes are characterized by having nucleus that separate the genetic material from the rest of the cell component. In the fossil record, eukaryotic cells do appear in about 1.5 billion years ago. Eukaryotic cells resemble to those found today. Evolved somewhere around 850 million years ago, and multicellular organisms made their first appearance only 600 million years ago. In multicellular organisms like human, different specialized cells perform different functions. For example, brain cell or neuron is structurally different from blood cell based on their different function. However, cells in a complex multicellular organism can be divided into two types, that is somatic cell and a gametic cell. Somatic cells are body cells and do not directly involve in reproduction. Somatic cell again can be of different types based on different function, whereas gametes are the sex cell which directly involved in reproduction. In human there are two types of gametes, one is egg, another is sperm. The egg is called as female gamete cell and the sperm is called as male gamete cell. Cell anatomy. Generally, eukaryotic cells share a similar structure. Every eukaryotic cell has a semi-permeable membrane called plasma membrane that bound all the components of a cell together. Inside the cell, all the organelles are float on a gel-like substances called the cytoplasm. The most important and prominent structure in the cytoplasm is the nucleus, which has its own thin envelope called nuclear membrane, separating its content from the cytoplasm. Inside the nucleus contain two complex molecules called as DNA and RNA. DNA are called as deoxyribonucleic acid is a double-stranded complex molecules, where genetic information is stored, whereas RNA is a ribonucleic acid is similar with DNA but have single strand. RNA is also found in cytoplasm. It is essential because it becomes the template for protein synthesis. Another important organelle found in the cytoplasm is mitochondria. The main function of the mitochondria is energy production, and hence is known as powerhouse of the cell. It is a capsule-shaped structure and carry its own genetic material called mitochondrial DNA or mtDNA. Structurally and functionally, mitochondrial DNA is similar to the nuclear DNA, but differ in the pattern of organization. And mitochondrial DNA is an important tool for evolutionary studies. It follows the mate RNA one line of inheritance, that is, inheritance from mother to her progeny only. This is because father's mitochondrial DNA does not involve in zygote formation. Another complex structure looks like a folded sheet called endoplasmic reticulum or ER is found in the cytoplasm. The main function of endoplasmic reticulum is to increase surface area within the cell for metabolic reaction to take place. Ribosome is another organelle within the cytoplasm which main function is protein synthesis. Ribosomes which are attached to the endoplasmic reticulum is called as attached ribosomes, and those found floating in the cytoplasm as free ribosome. This figure shows the different component of the cell like the plasma membrane that bound all the cell component together, the cytoplasm, a gel-like structure that fill the cell. All the organelles float in the cytoplasm. Nucleus, which carry the genetic material, it has a thin envelope that separate nucleus from the cytoplasm. Mitochondria, which is known as the powerhouse of the cell, located in the cytoplasm. Metabolic activities are carried out in it. It has its own DNA called mitochondrial DNA. Endoplasmic reticulum, it is found in cytoplasm, gives extra surface area for metabolic reaction. Ribosomes are actively involved in protein synthesis. It is present in the cytoplasm. Structure of DNA. DNA is the hereditary material contained inside the nucleus. DNA carries the inherited from parent genetic information required for life. DNA directs all the cellular function of an organism through protein. So, in order to understand the mechanism of genetics, it is necessary to know the structure and function of DNA molecules. The exact physical and chemical properties of DNA molecules were successfully discovered by two young scientists, James Watson and Crick, in 1953. Watson and Crick developed their model of DNA based on some of the properties of DNA which has already established by other scientists. In the late 1940s and early 1950s, biochemist Chargaff and his colleagues have discovered that DNA consists of 4 different nitrogenous bases, and these bases are distributed in a unique pattern. In a given sample of DNA, the amount of adenine is similar to the amount of thymine and the amount of cytosine to the amount of guanine. Another scientist, Rosalind Franklin and Wilkins, were also successfully able to produce an X-ray crystallography of DNA molecule clearly showing that the structure is helical. Discovery of DNA molecular structure have altered our understanding of biological and evolutionary mechanisms and completely revolutionized the field of biology and medicine. For their famous discovery, Watson and Crick and Wilkins were awarded Nobel Prize in 1962, but Rosalind Franklin dies 4 years before the award at the age of 37 years because of cancer. The structure of DNA molecules is double strands forming a double helix resembling a ladder twisting around its central axis. Each strand is made up of a chain of nucleotides. Nucleotide is the basic unit of DNA and RNA molecules. Each nucleotide is again made up of three components, that is a sugar, that is in the case of DNA it is deoxyribose, a phosphate unit and a nitrogenous bases. There are four different types of nitrogenous bases belongs to two classes, purines and pyrimidines. The purines are adenine and guanine and the pyrimidines are cytosine and thymine. Based on these four different nitrogenous bases, there are four nucleotides which can be represented by the initial letter of each base, that is capital A, capital G, capital C and capital T. In a nucleotide, each phosphate is bound to a sugar molecules and each sugar molecules bond with a nitrogenous bases. The nitrogenous base of a strand form hydrogen bond with the nitrogenous base of complementary strand, thereby resulting double-stranded DNA molecular structure. The bonding between the nitrogen bases followed a fixed pattern and it is always A to T and C to G, as it is possible to form two hydrogen bond between A and T and three hydrogen bond between C and G. In other words, the combination is between purine and pyrimidine. Purine are larger molecule than the pyrimidine and therefore this combination is necessary to maintain a constant distance between the two strands. For example, if the strand of DNA has a sequence of nucleotide A, T, T, C, G, C, A, G, C, the corresponding strand will have T, A, G, C, G, T, T, C, G sequence of nucleotide. So, if we consider the analogy of a ladder, the side of the ladder is represented by the chain of sugar and phosphate. Rungs are represented by the nitrogenous bases and bonds forming between them. RNA is similar to DNA except that first, it is a single strand unlike DNA which is a double strand. Second, deoxyribose is replaced by a ribose as a sugar in nucleotide. And third, pyrimidine base thymine is replaced by another pyrimidine base uracil and bonds with adenine. This figure shows the structure of DNA molecules. As the genetic material inside the nucleus are DNA and RNA, the chemical and physical properties of DNA molecules was discovered by Watson and Crick in 1953. The structure of DNA molecules is double helix, resembles a ladder twisting around the central axis. DNA is made up of a double strand. Each strand is made by a chain of nucleotide. Nucleotide is made up of a three component, sugar, phosphate unit and nitrogenous bases. There are four nitrogenous bases, that is adenine, guanine, cytosine and thymine. Adenine and guanine belongs to purine class, guanine and cytosine and thymine belongs to pyrimidine class. Okay, now let's discuss about the function of DNA. DNA, which is a genetic material. The genetic information to build up an organism is transmitted and stored as a sequence of nucleotide called DNA or deoxyribonucleic acid from the parent to progeny. So that it is the DNA which carry the information from the parents to the progeny. This complete set of genetic information for any organism is called as genome. So genome is nothing, it is just a set of genetic information. Genome itself does not perform any active role in the functioning of the organism. Rather it carries the information for producing protein in the appropriate times and depth. So this set of genetic information act as a carrier for the production of protein. So all the cell of an organism should carry the exact genetic information. In order to achieve this, DNA should be able to replicate itself